Now that we have learned about cellular transport and cellular energy production, a very important question pops to mind. How do we know what these cells do? In order to find an answer, we have to go back to the nucleus. When we talked about all the organelles in the cell, I told you that the nucleus is the control center. I also told you that the nucleus contains RNA and chromosomes, which store DNA. DNA, or deoxyribonucleic acid, is the hereditary material that contains our genetic instructions. These instructions are used for development and functioning of almost all living organisms. Nearly every cell in your body carries a set of the same DNA. In 1953, Watson and Crick described the structure of DNA as a double helical structure that contains the 4 nitrogenous bases, adenine, thymine, guanine, and cytosine. These 4 chemical bases we call nucleic acids. They store all our genetic code in info. If you've ever done some computer programming, you know what I'm talking about. Now, how does this work? DNA bases pair up with each other. Each base forms a hydrogen bond with another base on the complementary strand. The bases of the DNA have a specific bonding pattern. Here's a point you'll likely be tested on. Adenine always bonds with thymine, and guanine always binds with cytosine. Together they form a base pair. In letter code, we write this down as AT or CG. Remember these combinations very well. Let's say we have a strand of DNA, AA-TG, CA-TC, CG. Now we can predict what the complementary strand will look like. Since A pairs with T and C pairs with G, we would get TT, AC, GTA, GGC. If you think this is difficult, write down some random combinations and try to pair them up. You'll see it's actually quite simple. Okay, now that you've got that, let's move on. Because of this bonding, DNA is able to produce identical strands when replicating. This is a very important feature. I will say it one more time. DNA can replicate itself. This allows you to pass on genetic information to your kids and to the new cells your body generates during growth. More about this later. During replication, the 2 complementary strands are separated. This opens the bonds between the base pairs. A and T get separated and C and G get separated. Think of a zipper. An enzyme called reverse transcriptase now uses new A's and T's and C's and G's to fill up the spots of the old strand.